So our next speaker is Jan Ellenberg. I'm very honored to present him, and I thank you very much for your presence here today. It's really an honor and very important for us. Jan Ellenberg is a senior scientist and head of the cell biology and biophysics unit at the European Molecular Biology Laboratory in Heidelberg, and he is a coordinator of the Imaging Center of EMBL, which is a new open access user center for advanced microscopy. Very nice, by the way, really, really impressive. For over 20 years, he has been interested in cell division and nuclear organization, including systematic analysis of mitosis, nuclear pore complex structure and assembly, as well as chromatin organization and formation. His goal has been to obtain structural and functional measures of the required molecular machinery inside cells using quantitative 4D microscopy, imaging single molecule spectroscopy, as well as super resolution microscopy. His research has played a key role in large European-wide efforts on system biology for mitosis, microscopy automation, as well as unbiased computational image analysis. He has coordinated the European molecular biology efforts to make imaging technologies more accessible to researchers. I think most of you know the role that Jan has played in global bioimaging. I met him through the virtual meetings we started, and I was personally very impressed and inspired by your words and your efforts and your support to us. So for us, I'm sure for Lionel will agree, it means a lot that you are here in presence. So thank you very much. You're welcome.
Thank you very much. Thank you very much, Andres, and also Lionel for the very kind and generous introduction. Also, thanks to both of you for welcoming me here and hosting all of us. I think you did a spectacular job in hosting this meeting and making us all feeling extremely welcome and extremely integrated into your efforts. So thanks so much for this. What I want to do today is talk a little bit about the next thing that I'm engaged with on the European side after building community networks, because as we discussed several times today, that's a global effort that is still going on and is also still going on in Europe. And I had the pleasure of being in charge of coordinating Euro bioimaging, and then in the second half of that effort, global bioimaging for over 10 years. But in Europe, luckily now we have a community network and an infrastructure network, and several representatives have presented that here already. But of course, it doesn't stop there. There's more that has to happen to actually get the science done and go to the next generation. And so what I want to talk about today is basically what comes afterwards. And one of the activities I've been putting a lot of my time in the last 5 years was to set up a new kind of open access imaging center that actually combines multiple disciplines and normally not together used technologies in one place. And also how we use that as a model for driving ahead more quickly innovation and new technology development, because we will not be able to solve all the questions. We have the technologies today and we stay at that level. We will need new and more powerful technologies. So these are the two topics I want to discuss a bit, to give a bit of an outlook in the future and to maybe some thoughts for creativity. So before I do that, I want to connect my lecture to really the topic of this exchange of experience workshop, which is really tied to achieving the Sustainable Development Goals. And so here I want to very briefly just highlight where I come from. The organization I work for is the European Molecular Biology Laboratory. This is a 6-sided European organization. It's kind of the CERN for biology. We are supported by 27 countries directly in Europe and with associated countries in other regions of the world. And we're about 2,000 people doing 5 missions. And so I think what makes EMBL relatively unique and also put us in a good position to lead efforts like infrastructure access is that besides our core mission of excellent research, we have missions to provide scientific services from the 70s when we were founded and to work on technology development, innovation, and translation in order to be able to provide those services. So these missions are embedded in the organization that I work in. Now, at the moment, EMBL always works on 5-year programs, and our current program is entitled Molecules to Ecosystems. And I think that really links very well to the Sustainable Development Goals we've been hearing here from the UNESCO representatives and that many of you touched upon, because we really need to understand how life works inside its natural context. And that includes human life, but we are just one of many species on this planet, and we won't survive alone. So we really have to understand our health, but also the health of other organisms in their normal context at the molecular level to do something about it. So that means what we want to do with molecular biology is a very broad task. And that means life sciences will need a very different level of services and technologies to meet these challenges than we have today. We continue to have to work on this. So as part of its new program, EMBL actually is launching also new services to underpin this strategy from molecules to ecosystems. And so there are 4 of these areas of new services. The new EMBL Imaging Center that I will focus on today because I helped build it for the last 5 years. Mobile services that we can deploy in the field for ecosystems research. And a lot of those will also revolve around imaging technology, data science services, because all of this has to be integrated and put together, and specific services focusing on infection and microbial biology. So why is imaging so central if you have a molecules to ecosystem vision? And I think imaging is at the moment doing something that life sciences maybe have undergone so far 3 times in recent history. It's really a transformation of how we do molecular life science. So I would say molecular life science came to be with biochemistry. We understood what actually are the chemical entities inside living systems and realized they are kind of the same for every living system. There are always the same types of chemical entities inside. Was very revealing, but we didn't really understand yet how they were functioning. So I would say the next big transition was molecular biology to realize this is all encoded in the same substance, again, universal in all organisms, and we can change it. We can actually manipulate. But still, biochemists and molecular biologists traditionally focused on one gene or one protein or one aspect of this huge molecular complexity we have in every living system. So the omics transition in the 90s really, for the first time, allowed us to get a complete molecular inventory of living systems. And that is now like running water, but it's still a relatively recent technology. So we have pretty complete parts lists of most species on the planets, or are building those parts lists. But that doesn't mean we understand how they function or how they function in ecosystems. We really need to put these parts into their functional context in space and time. And that is what only imaging can deliver. It can understand the function at the molecular level. And so what we want to achieve is really to go from a living organism to the molecular understanding of how that living function operates rationally, so we can understand and fix it if it doesn't work properly, either in a disease context or because the environment is changing so quickly that evolution has no time to adapt to this. And this is going to be our problem in the next decades. Yeah, so we need this level of understanding. Now, how to do that? We think that requires infrastructure that is maybe a little bit unusual at the moment, because we need to marry structure and function technologies together and really bring these types of projects into one operational workflow. So what we have done with the EMBL Imaging Center is to not use only one area of imaging, but actually combine structural imaging technologies where you determine molecular atomic structure with functional imaging technologies where you observe living cells, living organisms in a functional context, and really bring that together. We know that's challenging. These disciplines typically don't work with each other and the methods are very different. So we have extremely high level support for expert scientists or expert engineers that help the people to do these technologies. And as was mentioned earlier in this meeting, we see this as a key platform for innovation and the next level of technology development. And of course, training is always an important aspect of providing access to such technologies. So that means we want to marry light microscopy and also other intravital imaging methods with the latest developments in electron microscopy. And actually, as I will say at the end of the lecture also with X-ray imaging technologies. And in between is a whole family of correlative methods. So you can bring the same specimen from an intravitro experiment all the way down to a structure experiment and really get in the same project an understanding about function and structure at the same time. So a little bit more detail, what do we do in light microscopy? It ranges from all the different super resolution microscopies to different modalities that then come closer to correlation. And then we go into correlative technologies where there is a very strong emphasis on cryo technologies, cryofluorescence and cryo-electron microscopy and robust preparation of samples, because only if natively frozen is the structure preserved for structure analysis later on, which then is the handshake to the electron microscopy methods where we take the vitrified cryofrozen samples, thin them if needed by preparing cryolamella, and then do high-end data collection for atomic structure reconstruction. And so again, we guide people through this entire workflow for individual projects with expert support. So the instruments are really key to the success of this and having good infrastructure of that, but it's not only the instruments, it's actually helping people upstream. How do I prepare a sample for these complicated correlative integrative workflows? And how do I interpret the data? Because often it's a first of a kind project. These are pilot pioneering scientific projects, kind of data that has never been analyzed before. So you really need a lot of help for the users in order to be able to do this. But that's the goal really of this center, to bring the latest research grade technology that is operating only in the best laboratories in the world to the open user community and to everyone who wants to come. So that's the team at the moment that does this. Top level engineers and scientists dedicated to support of people. And these are not EMBL scientists that are supported. These are external scientists outside EMBL that can come and visit and that really provide fantastic support to the users and help with their projects. Now, how about innovation? The technologies we've now put into the center, it's brand new. It's the latest cutting edge commercial and academically developed equipment. It will be outdated in 5 years. And so how do we work on the next generation of technologies? And so I mentioned EMBL has a technology development and innovation mission, and it has done that for the last 40 years. It has always innovated, especially also in imaging technologies. And these are just a few highlights from the last 40 years of what EMBL has done. And so our traditional model has been develop new technologies and then work with industry partners to make commercial instruments out of it. And ZEISS, as you can see, is one of them and is in the room here. But with each technology, we use different partners depending on where do we see the fastest way towards actually having an instrument that everybody can use. The timeline of operating like this before something is generally accessible is relatively slow. And this is not because the companies are not doing a good job, but because they have to do something that is easy to use and robust, and that takes time to develop. So we wanted to come to a model that is a bit faster than what usually happens. And just to illustrate what can happen, usually is an example of my own work, where I think this innovation cycle worked relatively quickly. We had a science project in my lab in 2010. My colleague Lars Hufnagel with us built a new microscope to support it in 2013. And there was a commercial product after 5 years on the market. So that's pretty quick. But still in this 5-year gap, you could have done a lot of research with this technology already before there was a commercial product. So how do we go to something that works more fluently? And so what we want to use the Imaging Center for is to do innovation in a more open context, where we constantly have new developments. They will happen also a lot in industry itself. Companies have very strong R&D pipelines inside their companies or in academia. There are extremely strong technology developments in physics and engineering that are far away from life science, and expose them as early as possible to a broad set of life science applications by putting them in a service environment, not putting them in an individual research lab that only works on one thing, but put them in a facility where 100 projects come in every year and where the technology can really be tested. What is it good for and how robust is it? So we think that that will really close the gap between a new technology and its impact in life science. It will bring new technologies much faster to life science research. And for those that develop technology, immediately say, is this good for life science or not? Is it maybe more suitable for other areas of research, so that we can move ahead faster? Now, the type of work I described to you, we do in the Imaging Center, from function to structure, there's not one commercial partner you can work with to do all of this. You need 4 or 5. And so our current partners are down here with whom we do this. And so we also want to establish a system where such innovation can be done with more than one commercial entity, because the future technologies will not come just from one company, they will come from many. We need to work with all of them. So it's a bit of a new model, bringing technology in before it's commercial, already expose it to life science applications, get life science into the core design of the technology development from the start, and also work with multiple companies together so that we can promote the types of methods we want to have in 5 years for our research. So that means fast tracking our own technology development in EMBL into service, but also fast tracking technology developments from academia and industry in general via exposing them to the research user community quickly into service provision. And the service provision will come with new feedback for the next generation of development. So this becomes a reinforcing cycle. We can speed up making technology accessible and providing the feedback that industry needs to understand, are these the right technologies for the community we want to serve? So that's the vision for the new center we opened at the end of last year. We want to make it a physical gateway to new imaging technologies. We can provide access for about 150 research projects each year. We hope to be able to go beyond that, but that's the number we say now because that's what we're actually doing now. In the first 6 months, we have supported 100 projects in the center. Also really promote technology innovation in this way by working with the leading academic and industry developers and become a hub for user access. On the European side, there are many different public funding bodies that support infrastructure access, but it's a bit complicated landscape. There's about 20 different projects you could get money from, and most users don't really know which one to go for. So we help them with this so they all can do their research for free at this infrastructure. Now, the technologies I described are maybe still relatively familiar to many of you, from electron microscopy structure determination all the way to intravital imaging. But actually, disruptive technology development in imaging is still continuing and actually, if anything, accelerating. We are getting faster and faster new powerful technologies. And one area we are looking at particularly at the moment are new intravital methods and X-ray imaging methods, especially with the new upgrades of the synchrotron light sources. What becomes possible there is incredible. And so that meant we needed also in Europe to think about how do we promote the development of these very different technologies together towards life science. And so that is something the European Commission has picked up. They have made a call for developing the next generation of imaging technologies to be used in research infrastructures. And so we decided to put together a consortium of infrastructures that will do this together with the leading technology development organizations in Europe so that we can really push these technologies ahead. So we are here in one Europe-wide consortium putting together the lead technology developers, the lead service providers, and industry partners in order to promote this general much more beyond the EMBL Imaging Center. And so the vision is really to be able to go from an ecosystem to a molecular structure determination. We have put together the leading technology developers, especially the big physics infrastructures as well. So the synchrotron radiation providers, ESIF in Grenoble, PSI in Switzerland, and also we have Diamond in the UK as part of this, where really a lot of X-ray imaging development is happening. And at the same time, the big centers that focus on electron microscopy and light microscopy in Europe to say, what are the next generation methods and how can we from the beginning develop them so that it's not just for one lab and for one application, but for the community in general. Couple that to the service providers. We have Eurobiomaging as a pan-European system for microscopy. We have Instruct as a pan-European system for structural biology. And for deploying those in ecosystems, we also have a marine biology infrastructure in Europe, EMBL. So all of these will provide this. And then we have many companies on board who say, we want to be part of this and contribute to the next generation. So this, I think, will be key to really develop new methods in an integrated way for life science, and at the same time already think about how quickly can we make them available to the broadest possible user community and deploy them also where they are needed. We discussed this at this meeting. One central facility cannot serve all the research needs. You've got to be able to bring technologies to where the research is happening. And again, this is inbuilt into this project that we will also deploy them in a mobile manner wherever possible. So coming back to the EMBL Imaging Center, this is up and running. If anyone is interested, just write us an email with what your project needs are and we will support. So if the way to Heidelberg is not too long, we are open to the world, not just to Europe, and support any scientist that has a project for imaging technology. So take advantage of it. Of course, 150 projects a year is a drop on a hot stone. So we can help some of you, but not all of you. And we are very proud to be part of Eurobiomaging. It's a node actually both in Eurobiomaging and in Instruct, the EMBL Imaging Center, where of course many more outstanding national facilities have open doors and also accept projects and support users. And that's really great. But I think we are also very proud that we are Eurobiomaging. We are part of global bioimaging because this is a global exercise. It's a global endeavor to set up life science as a community that we can deal with what the future holds. And I think to me personally, it's so much joy to be here at this meeting and to see all the communities that are successfully organizing themselves and are getting off the ground, and the world map that is becoming more and more populated with people with the same mindset that want to support this long-term mission. And this all started really because of Antje in 2014, because when we applied to the European Commission for the second round of support for Eurobiomaging, Antje said, okay, in my spare time over the summer holiday, we will also write a project for global collaboration. And I said, you're crazy, you're already 200% committed. Don't do it now, you will die. She did not die. We got the project funded from the European Commission for the first 4 years. And that started everything. Yeah, and that actually also started the Latin American initiative. So before Chris held up the flag for Mexico, it was actually Leah and Alfredo in Argentina that raised the flag for Latin America and said, you know, we also have a community and it is well organized. We can participate in this. And I'm really happy to see that with Latin America bioimaging, hopefully now everybody can come back into the fold and really profit from this, that there is a continental activity to bring people together and to get the governments to support that. Yeah, but of course, it went much beyond this. It's now really everywhere in the world. When the European Commission funding eventually came to an end, there was the famous valley of death of supporting community building and infrastructure. You have no funding. This also happened to Eurobiomaging. Luckily, I could convince EMBL to step in and say, okay, we are going to continue this. We need to pay the people that are doing this. They are doing a fantastic job. And of course, as you know now, since 2020, CCI recognized the value that this has and has stepped in as the third funder of this project. And I think this is a community that I think we need for the coming decades to come. And I hope we can get a lot more support for it than we have at the moment, by also really pointing out to our respective national governments that it is their responsibility to support life science better and invest in these community networks. So for me, the take-home messages maybe for global bioimaging are, this is from the community for the community. And I think that it's its uniqueness and its strength. In life science, you can't organize infrastructure top-down. You can't build one big accelerator for a couple of $1 billion and everybody's happy for 20 years. It doesn't work like that. Life science has to happen everywhere where life happens and where you need to study it. So you need a bottom-up distributed approach, because only the people who are close to the researchers understand what actually is needed in the next 20 years. So you have to build infrastructure in a different way. And I think the way it's done here, bottom-up, from the community for the researchers that use their technologies, is the only way. It will otherwise not work. Politicians are always tempted, let's just invest in one big machine and then everything will be solved. It's not how life science works. Yeah, so we need to do it like that. And I think it works fantastically. It is critical to enable life science globally to meet the societal environmental challenges that are coming up. They are going to be severe in the next 20 years. We need to be faster in reacting. It cannot take 30 years to understand one disease or to react to how do I mitigate climate change effects on the agriculture. We have to be quick in understanding this molecular and reacting to it so that we can maintain life on this planet. And that does mean the infrastructure for life science needs support in international collaboration that is not second to, for example, what happens in particle physics or astronomy. It will make a bigger difference for our life and survival than those fields, which are fantastic fields, but who are mostly academic in terms of what they are achieving. Life science is really about people's life and about the survival of this planet. It needs appropriate infrastructure to operate fast enough. We all have to make this case to our respective funders. So imaging technologies are key for life science, and the imaging community actually has, and I think will lead in many areas the way for how to organize, form the community and share infrastructure in an effective way. And I think you can all be proud of that because we are doing it slightly different than other communities, but actually in a way that works and that delivers. And that is a very good investment opportunity for private and public funders to actually enable the next generation of research. So that's what I wanted to share today, where we are going in Europe at the moment and where I've spent the last 5 years of my time. So I'm happy to answer any questions you might have. Thanks a lot.
Questions, comments? Don't be shy. There we go. Thank you for a great talk. The idea of linking, I guess, life cycle or sorry, life sciences into an imaging, which is obviously has a much wider role, but linking sort of life sciences into more sort of broader, should we say, context, like exactly the sort of the ecological and ecosystems and these things. Where did that come from? Was that something that was there a call to place things into greater context, or was that something that occurred to you and your collaborators that was missing, and that was something that could be extended too easily and was very necessary? How did that come about?
I think it's maybe there are two answers to this. So I think one is, a lot of in vivo imaging has exactly this goal, to observe the processes of life in their physiological context. Now, you can interpret that in a medical sense, that it's got to happen inside the human body or something that is similar to that. Or in a more broad sense, that if you want to understand a particular species, you would like to understand it in a physiological way. So just from a scientific point of view, I think this is the right thing to do. But the other motivation for this really was the discussion we have at EMBL. Every 5 years, what is the main direction of our research in the next 5 years? And that's a discussion we have among all faculty to say, what do we see coming? Where do we think are the important challenges coming up? And how do we structure our programs with which we then go to our member states? We have 27 countries that pay our budget for 2,000 people and tell them, this is what we want to do, and this is what it's going to cost. Do you want us to do this? And the last discussion we had about 3.5 years ago, everyone basically said we've got to be able to do molecular biology, no longer just in yeast, Drosophila, and HeLa cells, but we've got to be able to do it in the systems that are actually the most relevant for the ecosystems, and also focus on questions to understand how ecosystems are functioning, because they will break down and they will start to not have time to adapt to the environmental change. So this was really something that came from the faculty. Obviously in Europe also, the European policy process has identified such challenges as major societal challenges that investments should be going towards. Yeah, so it's I would say, a win-win situation between the scientific discussion and also the funders' agendas.
So real convergence,
yeah,
from a number of different areas. Yeah. Interesting.
I have just a quick question. So is the center, the new center, testing center, will that be open to people from South America to test also?
Yes.
It sounded like you kept saying EMBL and people.
No. So it's open worldwide.
Okay.
Of course, we would like to also support scientists in EMBL's member states, and those are predominantly in Europe. Although we also have non-European associated members. For example, Australia is part of EMBL, and Argentina for a period of a few years was also part of EMBL. Unfortunately, then because of the very difficult economic situation, Argentina stopped doing that. But we are open to people worldwide, and already now the first 100 users, we've got people, many users from outside EMBL member states. So the only thing we look at is the project doable, and we have a very short but independent peer review. Is it a reasonable project? So is it a good scientific case? But then anyone can come.
Thank you.
I don't have a question, but I have just a comment that I want to say, because what you say at the middle of your presentation today is something that for me is really, really important. And I have listened this from Leon one before about the importance of his center in Chanilia, and how important it is to put technology and technology development within a center who offers services. Because in some point, this is an argue that you have to deal when you have to, justify why you want to put a development, a machine that is developed in your house, about the impact that is going to happen. And this narrowing this distance of the developing and the impact of the technology is something that for me is the target. And now I am going to steal something else, which is what you say is how important is to basically put this technology under development, to test it, to be sure that this is useful, to be sure that you can modify as you need. So for me, your lecture today and Leon lecture are really, really inspirational, and I really want to thank you.
Thank you very much, Leon, for the kind comment. Yeah, no, I couldn't agree more. And that's really also at EMBL, this was not a research talk. We have 20 faculty that does technology development across the board of imaging, from data to probes to machines. And if you let them just do that, they will develop fantastic machines, but they may physically work perfectly, but they may not actually be very important for life science. And so that did happen at EMBL in the 80s, when we had a separate unit for just technology development, and they were kind of uncoupled from the biology units, and they did fantastic physics, but half of it was never used in biology. Yeah, and so I think we have changed completely how we do that. Now the technology developers are embedded in biological research units and in the service providing centers. So that really makes sure what they develop delivers biological research, and it's developed in a way that you already know it's not just for one project, it's for the community. We are trying to do something robust, something that addresses many things, and that can be used by many scientists. And so I think that puts people in a completely different mindset for their work. And also they appreciate it because they create impact much faster. Yeah, I mean, they can publish in much better journals, and they realize what I develop is needed. People love it. They are solving problems with it. I have impact. Yeah, so it's really a win-win for the physicists and for the biologists. And the way to link them is this service provision idea, that you really do this not with one project and one problem, but in a general sense. Yeah, so I think that setup, I think, is extremely valuable and really a self-reinforcing system. And again, something I can only recommend to think about when you set up facilities and you work with users, to use the opportunity that they sometimes come with projects that you cannot solve because it's beyond the systems you've got, to see that as an opportunity for new technology development and say, all right, we could do it if we invest half a year in the method with a company together or with an academic developer together. We can't do it now, but let's try to do it. Yeah, because the facilities are in a key role. They really know what the community needs. The individual developer doesn't. Yeah, so I think that's the key role that service centers really have. Andres.
Yeah, thanks for the talk. Also, I agree with Lionel that it's very inspirational. I'm wondering if you could give us an insight about how do you think when you envision a center like this, how do you choose which technology are you, because you cannot have all technologies, but you seem to have a lot, I mean, covering a very wide spectrum, but how do you choose, and particularly thinking about other places like ours, when we think about ideas like this and centers like this, how should we approach this
this problem? Yeah, I mean, so we've got a lot of people that work on imaging technology or use it at EMBL. So of course we talk to our colleagues and say, what do you think is the most important? So we get a lot of internal advice, and actually for the center we have an internal committee that meets every 6 months and provides this advice. So there's a constant process of updating this recommendation and looking forward. And we also get external advice from very high level people in the field to say, where do you think the future is, from very established scientists, several Nobel laureates among them, to tell us where do they see the needs in the future. But I think the most important is the user projects you get. Yeah, we now see the first 100 projects coming in, and people don't always ask us to do what we predicted they would ask us to do. Yeah, and so this really, in the end, you've got to ask the community what they need, but you have to offer what you can do. Yeah, and so I think this is the most valuable source of input, and this will evolve over time, and that's why we need to constantly innovate and constantly think about the next generation. So that's how we do it. But we will look at this every year and revisit it, and when we need to change, we will try to convince our funders that the investments for change have to happen. Yeah, so we were lucky now to set it up, that the support we got was very generous, and we indeed have a very broad portfolio of cutting-edge technologies, but that may not be enough anymore in 2-3 years down the road. Yeah, and we may need to change emphasis depending on the needs of the projects coming in.
Thank you.
So internal and external advice is our solution. We should stop. Thank you.